In this video, we're going to look at how bacteriophage T4 can be used as a model system for studying heredity. Mendel used pea plants as a model system. He crossed different pea plants and discovered the laws of heredity. Thomas Morgan and his group used Drosophila as a model system, and they used this model system to determine the first genetic maps. Drosophila have a few advantages over pea plants. They have a faster generation time, so we can make many crosses in a short period of time, which means we can measure recombination events that happen at very low frequencies and would normally require the analysis of many progeny. The drawback, however, is that complex organism phenotypes can result from the effect of a variety of genes, and we need to analyze a very large number of progeny, which can be time-consuming. For example, if the frequency of recombination is 0.001%, we need to analyze at least 1,000 progeny to find a single recombination event. In the 1950s, the founding fathers of molecular biology, Max Delbruck, Salvatore Luria, and Alfred Hershey, decided to use a simpler model system, the bacteriophage T4. These three scientists were initially physicists and thought they would have to discover a new law of physics to explain life and the transmission of hereditary information. As physicists, they sought the simplest possible model and chose the bacteriophage. If you look at the replication cycle of the T4 bacteriophage, it begins with the injection of its genome into the bacteria, followed by a large accumulation of new bacteriophages inside the bacteria. The bacteriophage genome codes for lytic enzymes that cause the lysis of bacteria and the release of new bacteriophages capable of infecting neighboring bacteria. The generation time of T4 is about an hour, which means that we can analyze thousands of progeny very quickly. However, bacteriophages are not visible to the naked eye. They're only visible with electron microscopy. So, we need a better method to visualize infection events and to detect progeny. Bacteriophages can be used to infect a plate of bacteria that grow on a solid, nutrient-rich medium. Growing bacteria completely cover the surface of the plate and form a milky opaque layer at the surface, called a lawn of bacteria. A small volume of the bacterial culture is mixed with a known volume of a bacteriophage solution. The mixture is added to a solution of top agar maintained at 42 degrees Celsius. Top agar is a low concentration of agarose in solution in a nutrient broth. At 42 degrees Celsius, the solution containing bacteria and bacteriophages in top agar is liquid. This solution is then quickly poured on top of a petri dish containing a solid medium made of a high concentration of agarose in nutrient broth. The top agar solution forms a thin layer on top of the solid medium, where infection events between bacteria and phages will take place. How is a lysis plaque formed? Let's consider an infection event between a bacterium and a bacteriophage. Here on the left, a clear zone in the opaque bacterial lawn is indicated by a red dot. In the microscopic view on the right is one bacteriophage infecting a bacterium. At a microscopic level, new bacteriophages accumulate in the infected bacterium, and upon lysis, the released bacteriophages infect surrounding bacteria inside the top agar layer. After several rounds of infection followed by lysis, a clear circular disc forms at the surface of the opaque lawn of bacteria. This clear disc corresponds to a region of the bacterial lawn containing lysed bacteria and is produced by a single infection event. The diameter of the lysis plaque increases over time because the number of rounds of infection and lysis increases. The number of plaques on the plate is equal to the number of infectious events and is used to calculate the titer of the initial solution of bacteriophage used in the Plaque Assay. In order to analyze such a model system, we need a phenotype that can act as an experimental readout. Mendel used the color and shape of peas, and Morgan used the color of Drosophila eyes and the shape of the wings. The phenotype that was used for these bacteriophage T4 mutants, called rapid lysis mutants, provided molecular biologists with a phenotype that is easily detected. Compared to wild-type T4 bacteriophages, these mutants have shorter infection and lysis cycles. We know that on plates, bacteriophage infection events lead to the formation of plaques resulting from infection and lysis cycles. The incubation time of the plate determines the number of lysis infection cycles and the size of the plaques. Therefore, on plates incubated for a given period of time, rapid lysis mutants form larger plaques than wild-type bacteriophages. As you can see in this image on the left, we have small plaques that are produced by wild-type bacteriophage T4, and on the right, we have large plaques that are produced by rapid lysis mutants during the same period of time. When using a plaque assay to measure the titer of a phage solution, it is essential to select a ratio of bacteriophages to bacteria that ensures single infection events. This typically involves using an excess of bacteria so that each bacterium is infected by only one bacteriophage. Accurate titer measurement requires careful quantification of the number of plaques formed, as this reflects the concentration of infectious phages in the solution. The titer of a solution refers to the concentration of infectious bacteriophages per unit volume, or the number of plaque-forming units per milliliter. Let's look at an example of how to quantify this. To determine the concentration of our stock solution of bacteriophages, we performed a series of dilutions. We started by diluting the stock solution, which we will call S0. We need to dilute our stock solution to create a solution S1 because the stock solution is too concentrated. At its current concentration, it will be difficult to distinguish individual plaques on the plate, as the high density of viral particles causes overlapping and merging of plaques. By diluting the stock solution, we can reduce the concentration to a level where individual plaques can form and be easily counted, ensuring accurate analysis and results. To create the first dilution, S1, we mixed 0.1 milliliters of S0 with 9.9 milliliters of buffer, resulting in a 1:100 dilution. Then we performed a second dilution, S2, by taking 0.1 milliliters of S1 and adding 9.9 milliliters of buffer, achieving another 1:100 dilution. This means that S2 is a 1 to 10,000 dilution of the original stock solution S0. Next, we took 0.005 milliliters of S2 and plated it onto a bacterial lawn. After incubation, we observed 250 plaques on the plate. To find the titer of the stock solution, S0, we back-calculated from this observation. We determined that the concentration in S2 was 50,000 plaques per milliliter. We know that we can calculate the dilution factor to make S2 from S1 as well. Once again, it's 0.1 milliliters in 10 milliliters, so it's 1 in 100. This means that the titer of S2 will be 100 times less than the titer of S1. The titer of S1 is 5 times 10 to the power of 6. Given the 1 to 10,000 dilution, the titer of the stock solution S0 was calculated to be 5 times 10 to the 8 plaques per milliliter, accurately reflecting the initial concentration. In conclusion, T4 bacteriophages are a good model system for studying heredity, similar to how Mendel used pea plants and Morgan used Drosophila. Its rapid generation time and the ability to produce large quantities of progeny make it ideal for genetic studies.